This is part 2 of Chapter 2's summary, The Chemistry of Life. In this section, we're going to review the 4 major types of macromolecules that are components of all cellular life. Cells are all composed of 4 major kinds of macromolecules. These macromolecules are used to build all of the organelles, all the components that a cell needs in order to accomplish the things that it does. The 4 kinds of macromolecules are lipids. Lipids are components that make up the exterior of a cell, that make up internal parts of the cell. Lipids are used to form membranes, and the main function of a membrane, as we'll see, is to keep things that belong outside of the cell outside, and things that belong inside the cell on the inside. That is, they act as barriers for the free movement of molecules from inside to outside the cell. Carbohydrates are components that are used to store energy and also typically will give structure to cells. Proteins are also present in a structural role for many cells, and proteins are also better known as enzymes. These are components that are used to catalyze chemical reactions. That is, they make chemical reactions occur on a speed time frame that is useful for a living cell. And then finally, nucleic acids. All living cells utilize nucleic acids as a matter for storing their genetic material. They are also components of nucleic acids are also used in the role of energy storage. This picture demonstrates the overall structure of the lipids. We can see the lipid is composed of a couple of components, composed of fatty acids, which are extremely hydrophobic groups, glycerol, which links the 2 fatty acids to the rest of the lipid, and then finally a functional head group, which typically has the element phosphate in it. The head groups are extremely hydrophilic or water-loving. So if we look at the elements that are used to make up lipids, what we'll find is that there is some carbon present, some hydrogen, oxygen, and phosphorus finally. Lipids have an interesting property in that half the molecule, the part composed of the fatty acid, is typically extremely hydrophobic, and the top of it, the phosphate-containing head group, is extremely hydrophilic. What that means is that one half of the molecule, the fatty acid component, repels water, and the top part of it attracts water. What this means is that many fatty acids will then associate, these lipids will associate tail to tail in a bilayer. We end up with a structure that is extremely hydrophobic in the center and extremely hydrophilic on the outside of it. Water then associates with the outside or the inside, but water does not associate with the center of this bilayer. What this means is that molecules that are also hydrophilic are unable to easily pass across the membrane. The second kind of macromolecule found in living cells are the carbohydrates. Carbohydrates are composed of sugars or saccharides. Individual saccharides are bonded together through the formation of a disaccharide bond. This is a dehydration synthesis, and growing chains of polysaccharides such as cellulose or amylose play structural roles in cells or play a role of storing sugar, storing energy for times when the cell may require them. The main elements that go to make up carbohydrate are carbon, hydrogen, and oxygen. There are sometimes some other elements that are small components in carbohydrates, but the main ones are these 3. The third major class of macromolecules found in all living cells are the proteins. The main building block of proteins are amino acids, and amino acids have the basic structure that's outlined over here in section A, with an amino group, or carboxyl or acid group, and it's the amino group and the acid group that give amino acids their name, and a functional group, a side group that's indicated here by the letter R. There are 20 naturally occurring amino acids that go to make up proteins in all living cells, and we can see some examples of the different types of R or functional groups indicated here in these greenish boxes in the top part of the figure. And it's actually the nature of this functional group that determines what amino acid it is. Individual amino acids are joined together through the formation of a peptide bond. It's another dehydration synthesis between the acid group of one amino acid and the amino group of the next amino acid in the chain. Amino acids are linked together through the formation of a peptide bond, which is a dehydration synthesis that we talked about earlier. Over here on the right, we can see a polypeptide, which is composed of 1, 2, 3, 4 separate amino acids, and we can see the R group of the first amino acid there, the R group of the second amino acid, the R group of the third amino acid, and then finally the R group of the fourth amino acid. The peptide bonds in both cases are going to be right here, and right here. And it's the primary structure that's going to be different for every single different kind of protein. Now, unlike lipids and carbohydrates, there are higher orders of structure that are very, very important for proteins. The primary structure, the order of those amino acids in the protein, causes the protein to fold up into a distinct shape, a distinct 3D structure. One of these higher order areas of structure are known as the concept of secondary structure, where stretches of amino acids that have a certain amino acid sequence may, for instance, have a tendency to form into a kind of a helical or spiral type of shape, whereas other segments of amino acids may have a tendency to fold themselves into sheets. The two main types of secondary structure that are frequently observed in different kinds of proteins then are given names alpha helices, which we see right over here, or beta sheets. We see some examples right here. And a large protein may, for instance, have segments of both alpha helices or beta sheets, or both of them joined together throughout the structure of the protein. Now, if we step back a little bit in scale and start to look at the overall shape of our protein, and many proteins can be very, very large indeed. Here we see an example of a random protein. It has a certain amino acid sequence. If we follow this little ribbon all the way around, it kind of wanders all the way around from one end to the other end. And what we see is that this protein, the primary structure, the order of those amino acids, has caused regions of the protein to develop into alpha helices and beta sheets. And these beta sheets and alpha helices all over the structure of the protein cause the whole protein to assume a very, very distinctive shape. And this overall 3D shape of the protein is what we refer to as the tertiary structure. The tertiary structure is going to be held together by hydrogen bonds, which we talked about in the first part of Chapter 2. These are very weak bonds that are caused by the formation, the association between hydrogen atoms and oxygen atoms that may be very distant from one another on the overall amino acid sequence, but because of the way the protein has folded, are brought into close proximity to one another. Now recall that hydrogen bonds are weak bonds. They're easily disrupted by the addition of energy, and so what that means is that this overall shape, the protein being held together by these weak bonds, it's also pretty fragile, and consequently the distinct shape that we see right here for this protein can be broken apart by the addition of something as easy as heat. Now, the fourth kind of macromolecule that we find in all living systems are the nucleic acids. The nucleic acids, such as DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are traditionally known as being the molecules that are important for genetics and heredity in cells, but they also play a role in the process of energy transfer in cells. And in fact, the molecule that you probably already know about, ATP, adenosine triphosphate, is one of the building blocks of DNA. We can see right here the basic building block of all nucleic acids. This is a nucleotide. It has a nitrogenous base, which is indicated right here, a sugar, and a phosphate. Nucleotides are going to differ in a couple of different places from one another. In the nitrogenous bases, and these are right up here, there are 5 different kinds of nitrogenous bases that are found in all nucleic acids: adenine, thymine, guanine, cytosine, and uracil. Nucleic acids also going to differ by the type of sugar that they find. Ribose is found in some nucleic acids, and deoxyribose is found in others. So ribose, of course, is going to be the precursor for making ribonucleic acid, or RNA, and deoxyribose is going to be the precursor for making DNA. You can see that chemically they're very, very similar to one another. In fact, they only differ down here, with ribose having a hydrogen right here and deoxyribose having an OH or hydroxyl group right here. The third part of all nucleic acids is this phosphate right down here, and the phosphate is part of the molecule that forms the link between nucleic acids as they join together, again through the process of dehydration synthesis. Nucleotides are linked together to form a nucleic acid where there is dehydration synthesis between the phosphorus of one nucleotide and a hydroxyl or OH group on the sugar of the next nucleotide in the chain. So we end up making a chain of polymerization reactions where a new nucleotide, triphosphate, is added to make a long chain of nucleic acid. So we end up with a long chain of nucleic acids which have been added together one after the other. If we look at deoxyribonucleic acid, we find that there are 4 letters, 4 nucleotides, 4 nitrogenous bases that are used in all DNA molecules: A, G, C, and T. However, if we're making ribonucleic acid, RNA, the nucleotide T is replaced with a U. So if we happen to look at any nucleic acid and we find a T in it, we know that it's DNA. However, if we see a U in it, we know that it's RNA. And one of the other properties besides this substitution of one of the letters is that DNA is typically going to form a 2-stranded chain, and RNA is going to form a 1-stranded chain. Now, this 2-stranded chain that we find in DNA is very, very interesting. This is the basis of heredity in all living systems. We find one chain of DNA, which I've indicated right there, and a second chain of DNA that's actually complementary in the sequence that we find on the DNA. So that every time that we find an A on one chain of DNA, the opposite chain is going to have a T. And if we have a T on one chain, the opposite chain is going to have an A. If we find a G on one chain, the opposite chain is going to have a C. And finally, if there's a C on the first chain, there will always be a G on the other chain. It's actually this complementary, this 2-chain structure that DNA has that's the answer to how this can be used as the repository of genetic information. Because when a DNA molecule gets copied by the enzyme DNA polymerase, what happens is that the DNA structure is zipped apart. These 2 strands are pulled apart from one another, so we end up with just a single strand by itself. And that single strand acts as a template for the formation of the second strand during the process of DNA replication.